Well, it's a pleasure to be here. Thank you, the organizer. This is a fantastic place to end this initiative, no? Contra. So I'm going to talk, otherwise we're going to be late. So as you may know, cancer is evolution, but evolution is nothing that has invented people in cancer. Neither people in cancer is leading research in evolution, right? So evolutionary biologists have been working for 200 years now or more, right? Understanding how species, populations, individuals, no, change through time, colonize different environments, move in space, and so on, right? So in my lab, we've been working through the years in different things. So it's not this, right? Working from island phylogeography with mollusks, crabs, understanding when they colonized X million years ago, the origin of HIV, where, migration, snails through Europe, expansion, colonizations, all this language is common to evolutionary biology about movement, about time, about space, right? And that indeed makes also a lot of sense for cancer, right? So formally, the study of the genetic variation in space and time is, sorry, because I need to get used to this, is called phylogeography, okay? Which is how we study historical processes that kind of limit or promote the movement through time of genes, right? There are barriers to this movement, which can be of any kind, physical, environmental, behavioral, of many different kinds, but that's something that we've been working with different models of different sophistication. And phylodynamics is other term that we use more to understand which are the processes that give shape to the phylogenies. Because in the phylogenies, in the genealogies, there is information, and that's the key message of my talk. There is information in the shape and the branch lengths of the genealogies that we can build. For example, you will see next for cells, right, about historical processes, which might be growth rates or movement in time and space. And for this, we use a variety of models. So phylogenetic reconstruction is just the beginning of the evolutionary analysis. It's not the last step, right? Well, in cancer, normally it's the last step. You do a tree, and then you look at it and say, well, no, you can do many things, right? Because there's information about processes, about evolutionary parameters. Like this, for example. So this type of tree either means that you have a lot of missing data, that happens a lot with single-cell genomics, or that is continuous selection, right? So there is a lineage, a clone, if you want, that is, replacing the previous clone and the other, so we get this kind of ladder step evolution. That's indicative of that, but I insist it can be missing data also gives this pattern, right? So through time, and that's what happened with HIV, intra-patients. This type of very short branches in the past are longer in the recent, in recent times. What we call the terminal branches is indicative of growth, right? So growth, where it makes the phylogenies to look like a star, with short internal branches and long terminal branches. Well, with constant size, we get the opposite. We get something like this, right? We get shorter and shorter branches as we go to the present from the past. That's the expectation. That's a mathematical expectation that we can formalize as being studied for a long time, right? And spatial structure, for understanding spatial structure, we need labels of locations. And we can have that in cancer. We can have labels for locations, okay? And that is the main point. We also, this is for, look at, for example, the intensity of the splits of the lineages, right? That we call skyline plots. Also tell us about the evolution of the population size. It's not the population size in terms of counting with your fingers. It's a little bit, it's a concept which is effective population size, right? It's under some models, some assumptions, of course. But the accumulation of splits through time, right? If analyzed statistically, can inform about a decline on population size or constant size, and so on. Sorry. Well, we've been using, and we will see that we have been using this for cancer, right? Models like these are for viruses. Most of phylogeography has been really well developed for studying viruses. And now with coronavirus, you probably even seen it, right? This type of things. But people have been studying viral spread using sophisticated techniques, right? And developing sophisticated software for analyzing the movement in a more or less discrete way, where you have the present and you have locations for your items, which might be cells or might be clones, and then you are able to reconstruct what was the ancestral location of the ancestor. Imagine these are clones, that they live in some location called E, then you can infer that this was an E or not. You can do a very naive model, which would be parsimony, which is what is implemented in Machina, for example. That's something that comes from the 70s, right? Or use more sophisticated models, like might be discrete, using Markov chains, Markov models of change, statistical models, likelihood, Bayesian statistics to infer the locations, right? Or using Brownian motion for more continuous phylogeography. The difference between continuous phylogeography and discrete is that you can infer ancestral locations which are not one of your sample locations. Okay, so you can put the ancestor in some place in the map according to some Brownian motion, and that's what people have been using to. This is the flu A, right? The spread through time and space, right? And well, we are trying to do this with cancer. Of course, you can also include, and that's very. Unfortunately, I'm not going to be able to show you results, but we are working on this also. You can also add to this in a statistical way as covariates environmental constraints for dispersal, right? In this case, for viruses, might be temperature, or if it's a virus that lives in plants, it might be vegetation, it might be whatever you want, okay, altitude, density of people, and in cancer, might be whatever you want, hypoxia, right? Levels of oxygen. Right? So I'm going to now use all the slides. So that's the advantage of being the first talk, the typical slides, right? I'm going to be fast. This all is being studied so far with, what we call germline evolution. Evolution for us, when we say evolution for evolution biology, is germline evolution. It's the evolution that happens through generations, from fathers to sons to grandsons, right? But we know that there is also evolution within our body, and that's somatic evolution. No? Since we are nothing but like 2, 4, 8, or 16 cells, there are already mutations taking place along our life, and sometimes we get some mutations that change the game and give some fitness to some cells that grow too much, right? And then we have a tumor, no? And along this place, of course, these mutations happen because of different reasons, right? But there are always mutations that happen, and accumulate, right? And we'll see more about this process. But there is a process of evolution within our bodies. Of course, we can represent, and that's also, from the, what is this, 76 is the paper, 74, I don't remember, no? Peter Nowell paper uses trees and phylogenies because evolution, that's the way we represent evolution, no? Like the lines of descent. In this case, the lines of descent is among cells, from a healthy cell to the first tumor cell you want, and then changes in colors, it means mutations that confer a different advantage to clones. Then some clones grow in size and some clones move to other places, to other ecosystems, right? And there are some selection, which might be immune or might be therapy. So this is the same thing that we see, and we've been studying with frogs or with bacteria or viruses. Same idea. The natural consequence of this process is intratumoral heterogeneity. Intratumoral heterogeneity is nothing else than diversity. Genetic diversity is population diversity, like in any kind, like our faces in this room. It's exactly the same, but at a different level, right? This diversity occurs, of course, between tumors in different persons, and also within tumors, and it can organize in different ways, right? It can be like what we call like a well-structured population like this, where it's confined, there are clear limits in space, or it's mixed. And we can study this using techniques already there. So the way I see, and many people see it, is that somatic evolution is just the continuation of germline evolution, right? So we have a couple of million years ago, the split between humans and chimpanzees, then we have the human lineage, then we have the current population of humans, and one of these humans has different cell lineages, and one of these cell lineages expands and makes a tumor. But it's a continuous, okay? Of course, there are differences, right? Because the main process is in germline evolution is meiosis, right? So there is recombination. It is that you form gametes and there is meiosis, and that makes a big difference because then it's recombination. Well, in somatic evolution is the main process of DNA replication, is mitosis, and there is no recombination in principle, right? And the scale, right? The scale is millions of years for this, hundreds of millions of years, thousands of millions of years, depending on where your scope of your investigation. Well, here is for tumors is years, right? So there are challenges. What are the challenges of studying somatic evolution compared to standard evolution? Well, this sort of evolutionary time, that was me, has a consequence, which is that at least for people used to work at higher scales, right, at larger scales, time scales, that we don't see many mutations. Okay? Even if you see thousands, these mutations are still segregating in the population. There are polymorphisms. They are not fixed, right? So that's a challenge for some of the most sophisticated models. There is no recombination, and this might be good for some things because things are simpler and allow us to use some models, as you will see next. We lose statistical power because when there is recombination, different pieces of the genome are independent. So they are independent markers of the same process. So they are effective replicates of the evolutionary process. In this case, no. So we have a large evolutionary variance. Okay, we can estimate parameters, but there is variance because we only have one replicate of the whole process, right, which is the whole genome in this case. Selection is assumed extremely important in cancer, right? We can discuss when or when not is important, but we know that it's important, obviously, at the beginning for sure. And there is a debate that recapitulates a previous debates in the 70s in neutralism and selection. Okay, that happened 50 years ago in germline evolution. It's still not, I don't know if it's solved, but what is true that most evolutionary models are neutral. In population genetics and phylogenetics, it's neutral because it's much easier. Because there is only one model of neutral evolution, okay, in which have some properties. But selection models, there are infinite models. So this is the null model, the simplest one, where you build your null hypothesis and then you can try to reject them, okay, for example. For selection, there are many selective explanations of something when there is only one neutral explanation of something, because neutral is a theory, is a model that has particular expectations. Also, in cancer, structural variants are important, while in germline evolution, they are mostly neglected. You know that a few weeks ago, the human genome was completed, and precisely these repetitive regions are the ones that were more difficult to study. And yeah, now we have more copy number variants, but that's not the main thing in evolution. Sorry. Oh. And probably one of the most complex one is that when we grab samples, the clones and the genes are mixed. Okay? That in population genetics is the equivalent of getting this, putting 200 banana, catch some 200 Drosophila flies here, the fruit flies, then squeeze them and sequence that. Okay? People does that in population genetics only for one reason, money. Okay? Only for money. Otherwise, we sequence one fly at a time. Okay? So here, that's equivalent of single-cell genomics, and I'll talk about that. But that creates a problem, okay? So this is one of the first seminal studies by Marco, Gerlinger, Charlie Swanton, about, with trees, multi-regional, to have trees, you need to have several things, right? Because there is no story of one thing, right, in this sense. A phylogenetic story. And you know that, well, they were sampling different times, different places, like this, right? And building a tree of these samples, right? But at the same time, we know that, and you might have read this, that we have criticized this approach of building trees of locations, because locations don't have a history, okay? There is no such a tree, right, of Andermatt, Barcelona, and London. They don't have a history. There is people that live in these places, that they have a history, but the places don't have a history themselves of colonization, because there is a lot of migration. It's very complex, right? So this conceptually is wrong. So you need to estimate clones, okay? The units of evolution are not the samples, because on Monday you sample here, and Tuesday you sample there. It doesn't make sense to build a history of samples, because they don't have a history, okay? Only clones. And I know that in the back of their mind of people doing this, they are thinking of clones, but the samples don't have a history. And that has implications about spurious things that you might infer. Okay? Samples in cancer might have a location only if they are what we call monophyletic. They have a single origin, like different metastases and a primary tumor. But multiple locations of the primary tumor, they don't have a history. Okay? No? This. So that, when I got into cancer, like now maybe 7 years ago, I was looking at this picture, and that looked more or less like we do with, I don't know if you remember the picture I put of some Cape Verde and conus snails. So it looks pretty much, right? So we can visualize cancer as something like having the main continent being the healthy tissue, and then there is like, a new colonization of a big island, which is the primary tumor, and then there are colonizations of secondary islands, which are the metastasis sites, right? And there is clones or cells, if you want here, these might be cells, with different genotypes colonizing and moving, all right? And that information is contained. If we knew truth, we could put colors to the branches, right? If we knew the true history here, and that will inform us about the movement, right, and growth of the tumor. And that would be tumor phylogeography. So we can try to do the same thing we do with viruses, with coronavirus, try to do the same in cancer. So how does this work? Okay, so this is our first sample, so I'm going to be brief about this because this is already published, but in this colorectal cancer, Joao was the main leading the analysis, so we got a bunch of samples, we sequenced them. At that time we were using ion torrent, but here it doesn't matter, on multiple places in the primary tumor, in lymph nodes, the liver, also multiple places. And we infer parameters here, right? So we infer demographics using these approaches I told you about, the rate of split of the lineages through time, okay, through inferred time. So we can infer, like at this point of size, which was when metastasis happen, like a big jump in size, a single origin for the metastasis. We identify some selective, we can pinpoint selection on particular branches of a tree using some methods. We give quantitative measures of time, of the tumor age, okay? We use like, you need to calibrate when you do time estimation, you need some calibration. Normally in evolution, we can use, for example, the age of a fossil, right? Or if we do island, but we are working on islands, we know that this island appeared like 20 million years ago. So we have upper and lower bands for the time of things, right? In cancer, that's more difficult. So we use like estimations from cell lines, for example, of colorectal cancer, of the mutation rate, things like that. And with that, you can calibrate your time. So we estimated this tumor age is around almost 7 years old. No? The growth rate, we also measure, these are the posterior distribution of these parameters, and also migration through time, right? So in this case, we were able to infer, probabilistically, of course, truth is unknown. This is our best bet about what happened through time, how there was movements, in fact, in this case, back and forth of lineages jumping between the main tumor and the liver. So for this, we use a particular type, if you want, phylogeography, which is biogeography, which is more for species instead of populations. Why? Because here, here we estimated clones using a clone deconvolution algorithm. I don't know if it was Litchi, probably. Okay. So one of these ones, we use several, but two of them kind of agree. So we got clones. These clones are noisy, of course. They might be true or not. We don't know. But we use these clones, the sequence of these clones, right, which are these letters here, to infer their history and the location of the ancestors, and then we can infer movements. And here we use methods which are called biogeography because a clone can live in multiple places. So that's a big difference, okay? So here we infer the range, the geographical range. So the states of these nodes are the range, not a given location. Is in how many places do you live, right? For single-cell genomics that we'll see next, we have to change a little bit this model, right? So in this paper is a paper about clonal phylogeography. Okay. But clonal phylogeography, which is this, is that what we did in that paper, is a representation, but it's a summary, because a clone is a summary of the genetic variation. What is a clone? We could discuss, right? I'm thinking of a clone here of a main genetic lineage. I'm not talking about function. It's just a main genetic lineage, okay? But it's a summary of what is inside, right? Because there are more branches inside each one of these clones. You might say they're identical. Well, depending on your scope. Identical, there is nothing identical if you sequence the whole genome in a cancer. Every cell is different, okay? So a clone is also lack of resolution, right? Depending on your depth and target of your genomic study. What is true is that inside these clones there are other lineages, and we want to use that information of these lineages, because I told you in the first slide or in the second that we use these branch lengths to infer parameters. So ideally, we will move from the clone phylogeny to the cell phylogeny, okay? Because then we will have the full lineages. Of course, it's a sample always. We're talking always of a sample, indeed, okay? And that's where we move on in our lab, right? So we started. You know what is single-cell genomics? In this case, Well, instead of getting a bunch of cells and sequence all of them, and the data is more or less clean nowadays, right? We isolate one with some method, right? In our case, we use FACS. Okay? And then we need to amplify most methods. There are methods that avoid this step, but these are more or less in-house so far. And you need to amplify your DNA. And when you amplify the DNA, you introduce biases, right? In the data, and the data is more sparse, is more complex, right? And the biases are due to the heterogeneity of the coverage, because when you amplify some regions and you don't amplify others, and the cell also has lesions, right? When you isolate the cell, so you will have false positives and false negatives like this, right? So you're going to miss some alleles out there, allele dropout, right? And you're going to infer some mutations which are not true, either because there are lesions that happen during the isolation of the cell or during the amplification, because you need to replicate the DNA in the amplification step. And then you have a matrix like this instead of this, which would be a clean one. You have like a matrix with missing data, with errors that you, of course, don't know there are errors. You need to infer them, and so on. So we started like doing some experiments also with cell lines and to see the properties of the different kits of the amplification, and you will understand why. So we study many properties about allelic bias of different kits for amplify, correlation of the reproducibility of the results, heterogeneity of the coverage along different chromosomes, and blah, 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 because we wanted to select something like this. We wanted a kit that would always make the same mistakes, that would try to amplify the same regions recurrently, okay? So we prefer to miss 50% of the genome versus a kit that missed 20 if the kit I choose always amplify the same regions. Why? Because to study the phylogeny, okay, of a set of cells, we need homologous regions. We need to study the same region in all the cells, right? So we prefer kits, amplification kits, which are recurrent in the way they amplify the genome. And that's the case of Ampli-1, right? And it's not the case of Repli-G, for example, which is the most common. So
can you repeat the name of the...
Ampli-1. Ampli-1 is a kit which, for example, look at this, the recurrency for 10 cells. This is the number of cells that share coverage for some site. So up to 30%, this is whole genome, okay? Whole genome sequencing, like 25% of the genome is shared by the 10 cells. And that's nice, but other methods, this is more flat. So, or even like this, okay? For random methods that they are not recurrent in the regions they miss, are worse for phylogenetic reconstruction. They might be better for other purposes, but for evolutionary purposes, we want to have the same regions amplified. Even if we always miss 30% of the genome or half, we don't care, because if we are doing whole genome sequencing, we will have enough sites. Well, okay, so now we know which kits we need to use for phylogenetic reconstruction with cells, or single cell genomic kits, and also we realize of something, right? Is that I mentioned that somatic evolution is through mitosis. And mitosis is an ordered process. Well, there are not many biologists here, right? But mitosis is the process by which two cells divide, right? And meiosis is the way in which gametes, okay, sperm cells and eggs are formed. And they are totally different, okay? Because in meiosis, you need to split, you need to form from diploid, haploid gametes, you need to split your genomes, and you need to recombine them to generate diversity, okay? In sexual organisms, that's the advantage of sex, in fact, recombination, okay? Because it creates diversity. But in mitosis, it's perfect. You need to have two identical copies, okay? So it's order. The segregation of the chromosomes is order. It's perfectly planned in principle, okay? So they are identical, the copies. So the history of the maternal and the paternal chromosomes are identical also. And that's very important because otherwise we couldn't use models like this. So the standard models we use in phylogenetic reconstruction often for describe the changes from A to C, A to T, A to G is a 4x4 matrix, okay? But in this case, our data is not A, no? In cancer, the way we sequence, the data is AA, AT, AC, okay? For some people, you can summarize this as 0, 1, of course, but that's losing information about the identity of the genotypes, right? So here, what we did is to adapt the 4x4, matrices that we typically use in, to study frogs, to study cells with 16 states or 10, depending on whether you consider the phase. So we implemented a model for both cases. This is because it's faster but here is the 16 states, right? And we don't know the phase of the genotypes normally, right? But we can integrate that statistically. But with likelihood, we can see this is an ambiguous state, okay? So we give the same likelihood to both phases. You understand what is phases, no? Being in the maternal or paternal chromosome, right? So we can deal with that. So we build also like a genotype error model, which is a little bit different from the previous ones, no? Based on parameters like allele dropout and amplification rate. Amplification error, which is all the errors that happen also before the isolation or during the isolation, right? And then allele dropout. So this is the consequence to deal with the observations, right? For us, the observation can be also the likelihoods. So you have a color that gives you the likelihood. We can use that instead of the genotype of the best genotype. We can use just a vector of 16 likelihoods to consider uncertainty in the calling process, and that's very nice. So with some calls, we adapted a very popular. So there are software tools in phylogenetics that they are being used by 100,000 people, for example, okay, to get you an image. And RAXML is a program for phylogenetic reconstruction that also has thousands and thousands of users. So what we did is instead of writing a de novo program, I talked with Alexis and Alexei, Alexis Tamatakis and Alexei, which are my colleagues and friends. So we introduced the modeling in that software, and that software is already being tuned through years to be fast, to working with all kind of threads, OpenP, MPI, is optimized for calculations, right? So it's already published now. You can check it, and it's a maximum likelihood method to reconstruct phylogenies that works very well in simulations, and that we've been using to build our trees. Okay, and you can more details in the paper, right? Senbai is going to talk about extension or improvement of this. Of this method, which takes into more uncertainty into account, right? So we apply it to data like cancer data, of course, but that's going to be next. And then this is like hematopoietic study, maybe for Henry Lee Six and Peter Campbell and so on, and we study there. And nicely, our method, this is very simple, but nobody does this in cancer. I don't know why. We can provide confidence, right, using techniques like bootstrap of confidence on the internal nodes. So we can distinguish that this study, we repeat the analysis, and it was very well supported. You don't see here the numbers, but they are very high. The value goes from 0 to 100, 100 being a perfect support, okay, of the nodes, of the history. And this data is very well supported. And in fact, nicely, our program estimates a 0 ADO. And that's fine because these are single-cell colonies. So they got single cells, and they expanding into colonies. And then they sequence the colony. It's bulk sequencing, and they use the clonal mutations, which are the ones that are in the cell that was expanded into a colony, right? That's the perfect way of doing single-cell genomics, but it's very expensive and time-consuming. And you can distinguish that from this other paper, where this phylogeny is totally random. So the fact that you can build a tree with whatever. You can build a tree with plates of cars, right? With whatever you want. That doesn't matter. It has a meaning, a tree. So we need to be careful about that. So this tree is unsupported by the data. Okay, there is a bootstrap of 72, and here all the values are below 50. Values below 50, normally we don't plot. So this is random. The fact that you can build a tree doesn't mean it means something. This phylogeny is not supported by the data. Therefore, you shouldn't use it for anything, okay? This is for neurons. We also apply it to previous data to compare. For example, this is in paper in the Nick Nevin lab when they propose like a late, I don't know why they say late because there is no time information in this tree. That's an example of a misinterpretation. There is no time here. The fact you see a branch here and there, that doesn't mean that this happened, this happened before this. No, there is no time information there. That's a misinterpretation. But they propose 2 metastases. Well, with our tree, does it matter to use a different method? Well, we obtain like a single metastasis. And there are the studies also confirming this. So the method matters. It might have biological implications, okay?
Sorry, can I ask a question?
Sure.
So can you explain why, if they have such a tree, and then if I interpret that tree, there are some nodes which are like lower down
Yep.
and upper, why doesn't it mean that it happened earlier?
Well, because this goes against my nature. Because these mutations, you don't know when this mutation happened, or this one. Maybe all these mutations happen, in year 1, 2, 3, and 5, and this mutation only happened 8 years later. There's no time there.
Between the branches, we know that in the trunk that was earlier, right? But in the between the branches.
So you know that this mutation happened. This happened after this one for sure.
Yeah.
But when there is a split.
Yeah, exactly. In
yes,
the branches.
okay. So between this is only like a partial order. I cannot order between the.
Yeah. So this one and this lineage.
Yes.
You don't know the time, the relative time among them, right? Of course, this happens after this, but this one maybe happened after this one. We don't know it really.
Okay. So yeah, I misunderstood what you said about the.
Oh, sorry. No, I meant this. But they got confused about this. They think that this has to be somehow before this, and we don't really know this.
Yeah.
Okay, so we're analyzing now a bunch of data sets that we produce in our lab. Okay, don't do this. So I open a wet lab, having no idea of being always a computational biologist. I opened a wet lab 5 years ago for doing single-cell DNA sequencing, because RNA sequencing is a piece of cake, right? RNA sequencing, you have 2,000 machines that do it for you from the very beginning. And of course, you have many RNA copies in a cell, and DNA you have 2. DNA sequencing has been abandoned, in fact, unless it's for low pass copy number variation, where you have machines also for this. But for standard SMB phylogeny, that's what we need, SMBs, right? It takes a lot of time, a lot of error, a lot of trial, a lot of problems, and well, so it took us years to get this data. Now we have it finally, and we get, nice number of markers, if you want, or sites, like thousands of them, right? Which are enough to do some more complex likelihood of Bayesian estimation of parameters. We're going to do this with dozens, okay? So that's the data we have, and we are starting to analyze, and this is the type of trees that we are getting when there is blue is healthy normally, and colors are different locations. You can see in some patients, there's a structure. This is our most complex patients in the sense we have many samples and many different organs and the others. And we are now this might be the end of the analysis, no? In a standard cancer paper. Say, okay, these are my trees. But that doesn't mean anything to me, just this. I can pinpoint things with my eyes because I've been looking at trees for 250 years now. No, sorry, 30 maybe. So, yeah, there are things we can spot here, given the branch lengths and given the colors, but we want to perform analysis on this, right? And we also can do some detailed genomic analysis, like putting mutations on branches, right? And we can infer things which make sense, which is nice, right? Using single-cell genomics. In this patient, this is our patient in our lab, where we have the common usual suspects of colorectal cancer, mutated, which makes sense. There is like a tendency, if you want, these are locations, right? The shape and the colors. Also, we have other things like cell phenotypes. That's another story. And then we can map the mutations. Of course, in the trunk, we have the majority, and we have a support, and then we can infer open selection. But we can do more than this. That's this description. This is like a genomic description. We can start to look at things like we will see next. No? This is like another one, a little bit more complex, when we have like an independent benign tumor. Okay, you can see in the history, these are the healthy cells. There's two independent tumors, one that got malignant, and this is like a benign tumor that also have a history. But the support is perfect of the metastasis. Here, the story is very neat in this case, right? And now we have sorry, we couldn't finish this analysis. We have immunohistochemistry data to measure oxygen levels, to measure lymphocyte infiltration, and we are correlating, this history with the environmental constraints, and that's what I want to do. This is this phylogeny. We estimated some parameters here. This is like this is Bayesian posterior estimates of the location of the ancestors. And you can see that the history is quite simple, like a few movements here, changing from here to there, from this location to that location, and from here to there. The color is the region, right? So, but here we have two independent migrations, right? And we also estimated, this is like a Bayesian covariate. You can do like a GML analysis here on top of this, and we can see that there is a significantly more movement of the cells which are in a metastatic site versus cells in a primary tumor, which makes sense, but we can see it with single-cell genomics data, and we can measure this, okay? It's significant. This rate of migration within METs is much higher, this is the base factor, than between primary metastasis and within the metastasis, right? So it makes sense. Once you acquire some properties. Of course, you have to do EMT, to change your affinity, your addition to other cells. You need to become free, but then you need to grow again. But these cells are prone to move once you are metastatic. This is nothing new, probably, but we can see it.
Can you elaborate a bit on this? What do you mean by rate of moving? Is it like
changing locations?
But do you have the location of the cells within?
The location of the cells is because we sample. No, we don't have a location for each cell.
But you have like samples from certain spaces.
The colors are locations. Healthy cells. This is liver. These are nodes, lymphatic nodes.
What do you mean by moving? Because you have the cells in a certain location, and this is just to verify, I guess, that you can split the tree according to the location.
No, moving, I mean that one ancestor was in one location and the descendant in a different location. It's not the moving of these cells, it's the history.
Okay. Well, is a location a metastatic site, or is it also within?
No, all these, here sorry, we don't have a legend here, but no, these are, the number is different. This is the column. So we have location 32, location 40, location 42. I don't know if you see the numbers. So these cells, these 3 cells are in the exact same location. This is another one, this is another one, all within the primary tumor. So the color is the region.
But, if you compare with phylogeography, and, there were ducks living up on Gemstock a million years ago or so. Gemstock is the same thing, right? But locations in the primary tumor is sort of much more plastic, right? Because everything can move.
The location is defined by the pathologist, the way that, is the best you can do. No? I mean the best. It's not the best. It's
right, but you can be at a certain place because,
no, but place.
Other cells moved you there.
No, but the location is a more broader scale. It's not the scale of, it's in the organ. No, you can define. I don't know. It's macros. It's not like exact position. You can be. You can find locations. Uppsala and Stockholm can be different locations, or Sweden can be a single location. The whole of Sweden is dependent on your scale. So
right.
That's what we do.
So what I'm saying is that in a primary tumor, the location is defined by the population and not by a geographic sites.
No, for us, the location is the sample site. So the pathologists take a picture. I say, I grab. Well, the locations are already, sorry, because the locations are these. We have in the picture, oops, sorry. Oops, I'm gonna be late, no, like typical. We have a picture here, no? When was this? These are the locations. So this is one location, this is another location. Yeah, inside here, there can be movement, or that's at a sampling time. This is location 33, and this is location 32. Do you have a problem with that, or?
No, I'm just saying that, the fact that a clone is here and here may be because there were cells popping in between the cells of that clone, right? And sort of forced them to move apart. So that's something very different from a duck moving from Gemstock to Zurich.
Well, I'm talking, here there were clones, but what I'm talking now is cells.
Yeah.
So a cell is either here or there. But there was some reason for movement. We don't know the reason, but we can decide there was a movement. You know what I mean? It's like, I don't see anything wrong with doing this.
I don't see anything wrong,
Ah.
but there's a difference which may be interesting.
Okay, we can discuss this later if you want. Otherwise, I'm going backwards. So,
Wrong direction. Well, it's like there were already some questions, but you have like 5 minutes left.
yeah, because I'm not going to be able to finish. Okay, so we can estimate parameters. This is a study with Sulafa. You might know her that she was in Cambridge, I think, no? So this is like a story about only on metastasis. So we have like this liver. We got 2 samples from this liver. It was resected. There was some chemotherapy, and then we got there was a relapse of the metastasis in the liver. The same relapse or not? That was one of the questions. And we have 3 regions, and we have some clinical data about this. So we sequenced 60 cells, if I remember well, whole genome. We got almost 40,000 SNPs after curation. Takes a lot of time to curate the data also, right? And then we got our inference in which we have the before relapse and after relapse population. They are perfect, they are beautiful. This is the history, makes sense totally. We have the runs of chemotherapy, this is the resection, and we can infer the population size. This central part is extrapolation, really. It's not, it's a statistical extrapolation. Of course, we can only infer sizes when we have splits, really, here and there. But the history makes sense overall, right? So there is like, once there is the liver is resected, there is a demographic decline, and then when it appears, it's growing, right? So this is an extrapolation, which might be true or not, but we don't have much data for this central part. But that's the demographic history after this. That's what you expect, no? After the resection, the tumor size needs to go down. So, but this is inferred only with genetic data, okay? Of course. We also infer some interesting things that this is we can measure the substitution rate, and the colors is the intensity of the rate. So we have like a cold evolution time. After the resection, it looks like these cells were very slow. They took them a long time to grow. And in fact, here we infer, in this relapse branch, like a DNDS ratio, which is the statistic we use to measure selection normally. Like, it's not significant, I acknowledge this, but it's, before that, there is positive selection, here there is negative selection. It seems like, probably chemotherapy, or it's just the environment, we don't know. After the resection, these cells don't grow and don't change. They don't acquire non-synonymous mutations, in principle. There is something preventing them to change. And Monica did some analysis of the signatures and to see what's going on, right? And we found some well-known signatures that are related to or linked to chemotherapy, 5FU and platinum there, occurring during this relapse branch, right? So somehow the therapy is making, several things. Okay, it's preventing these cells to grow, but at the end, the cells end up growing. But that's a nice story, I think, and an example of doing this. So I'm going to skip this probably. This is something we're doing with mouse. Yeah, I know that I got into trouble opening a wet lab, and now I'm getting more trouble working with animals, but hey, I guess that's the nature of the beast. So we want to understand what is going on with circulating tumor cells. This is our current project. Where they come from, what do they do, right? Whether they, you know that CTCs make clusters, and clusters are 50 times more metastatic than single CTCs, right? And there is the hypothesis that there is clonal cooperation. Of course, if you travel together with friends, that includes neutrophils or things like that, and you go to a new place, and there is diversity, our hypothesis, these are diverse, it's easier to grow, because then you have different capabilities, right, in a cluster versus a single cell. So we're studying this question with some experiments, with some colleagues, of course, doing mice, but we have some very preliminary data, right, here about M is metastasis, and these are, sorry, because this was cells, single cells, clusters is in purple, and then we have metastasis, and here what we can see is a list of the origin principle of the clusters and the single cells is intermingled. That doesn't mean that there's a particular lineage of cells that become clusters and others that become single CTCs, no. It might be a question of chance and location. We think that clusters form in the tumor, and then they kind of lose adhesion, and they go to the blood or wherever, or to the lymph together. But there is also other theories that say that they get together in the stream, right, in the blood stream, which might be more difficult, right? Here we'll support both ideas, right? But this is on the works. And we've been doing also, Monica's going to talk about this. Fabian is going to talk about estimating functional clones with genetic data, okay? So we built this model about using this, something to what I just mentioned about these parameters about growth and the branches and using coalescence theory. So Fabian, together with Karsten Biouf, a mathematician in Copenhagen, we built a model, a very sophisticated model, that in fact will allow us to identify multiple clones with multiple growth rates, right? So this is allowing the tree to have multiple change points for growth rates inside. Okay, and Fabian is going to give you more details about this model. And well, we are collaborating with more people in CONTRA, as of course, with Eva. Here in this case, this is work in progress that Semba is going to present next, and Nico also. And this is the take home, okay? So we can have statistical phylogenetic models that we know them already. We've been using and apply them to cancer. We need to adapt them and make changes, of course. But I think that we don't need to reinvent the wheel, okay? Because people have been thinking of this for a long time about models for evolution, right? So we need to adapt them. I would say that that's like faster. We can obtain meaningful insights, right? The noise in the data is a problem in single cell. It's a problem. We need to be very careful. I didn't talk much about this today, but to get these matrices, you need to make decisions, okay? And I like this word, quantitative understanding. I like parameters. I don't like to say move to here to there. I like to see 28, 44, 0.8. That's important. I'm a biologist, but some biologists, we care about numbers, okay? And of course, thank you to a bunch of things, to Contra, of course, and funders like usual, and collaborators and people in my lab, and of course in Contra, people doing secondments, people that we didn't do secondments, but we've been discussing in these meetings, okay, and people that we are submitting papers in Contra. So thank you very much.
So we have still like 5 or 4 minutes for questions.
Oh, that was more or less good, the time, no?
Yes, yes.
So for the trees, when you show the locations, the one that was causing problem to you,
He didn't know problem, no? But
I'm wondering, did you ever saw metastasis circulating back to the primary tumor?
yeah, in the clonal paper, in this paper, the first story about clones, we saw or infer. I mean, when we say we saw, we infer, right? Retrospectively, back in time. So this is like truth is unknown. We infer like some cells from the liver going through the lymph nodes, back to the lymph nodes of the colon. You know, there is a hypothesis by Xavier Massaguet, it's called the self-seeding hypothesis, about that. There is attraction, right? Like the primary location, you know, attracts cells back. And that can be studied because a postulate of that hypothesis that these cells are going to occupy the external part of the tumor when they come back. Of course, they're not going to get into that. Our data supports that, but we don't know how general is this, right? We have no idea. But that's possible. Why not? I would say, no? Cells move. Once they are metastatic, they move. So most tumor cells, most circulating tumor cells die, but the vast majority. Most circulating tumor cells that reach an organ die. Only a very few are able to complete the process and create a new tumor. But that's maybe, I don't know, 1 per 1,000. Who knows? But that can happen. After once you grow, you are a malignant cell capable of growing in a different place. So you have capabilities for moving and colonizing other things. So why not come back to the same? And this paper by Xavier Massaguet is about segregation of cytokines, if I remember well, things like that. It's about, there is some biochemistry attraction for the cells. So biochemical attraction. Eva, I think it was first, sorry, but.
Okay. I had a follow-up question about these circulating tumor cells. Also, did you see, like, between the metastases that was also reported a long time ago?
That's part of our experiment. Sorry, I didn't have time to show this, but we have two main experimental designs. In the second one, we are carrying the primary tumor of the mouse. So it's going to have metastasis. So after a few days, all the cells that we find in the bloodstream can only be from metastasis, because they die very quickly, no? the cells in blood. And indeed, it makes a lot of sense that once you're metastatic, you have capabilities that, you know, we don't have specific numbers. We are going to try to measure them. How often CTCs come from primary tumors, from metastasis, and how often the migrations are between metastasis and within. Well, in that paper, in that manuscript I showed you with Sulafa, yeah, we do have this estimate that there is much more movement between metastases than anything else, which makes sense. And that suggests that circulating tumor cells, which are the main actors of metastasis in principle, right, they move through metastasis. So metastatic sites, they contribute circulating tumor cells. Yes, this is known. They do. They don't only come from the primary tumor. But the proportions have never been measured quantitatively. And it's only with mouse models. So we can only do this with mouse models in principle, right? Because we can do these experiments. We can remove primary tumor and see what happens. And that's what we're going to do, is plan to do this. So we will have some numbers, I hope, next year, maybe.
That's the last one.
My question is on the multiple sample stuff that Jens was asking. So did you see like the samples which are close spatially, were they in a similar branch or?
Yeah, there is, it depends. That's what we call population structure. And we do have, so a very simple way of testing this is comparing, you have two matrices of genetic distances. So you have like a square matrix, like a triangle there, and then you have a matrix of geographical, of genetic distances, and then you can do like a Mantel test, for example, something like that, whether these are correlated, and depends on the pacing. Some cases there is this structure, and some cases it's not. So when there is no structure, it implies movement somehow. But so, as you know, cancer is very miscellaneous, and every tumor is different, so every story is different for every tumor. So I don't think we have rules for this, in this case, for the structure. That depends on many factors, including chance, I'm sure, no? For the yeah. We see, in different patients, we see different things regarding that question, but we can measure them easily. We have statistics already for this called FST, for example, which is that comes from 1903, okay? The fixation index. So we have, that's standard population genetics to measure whether you are more a genetic neighbor of your geographical neighbor. But we see all kinds of cases in that regard. So.
Thank you.
Thank you.
That's nice.